many areas of the body, and they contain heparin as one of their granules, and we'll see that that's anticoagulant. We do have some smaller mast cells that line the digestive tract, and so they're there as well. This is an electron micrograph showing the nucleus, and I think we found making it dark was maybe preferable. It encourages nap time, but maybe this will be all right for us. This is the nucleus with the heterochromatin and the euchromatin around, and then in the cytoplasm, it's just really full of these granules. The ones in the digestive tract, instead of heparin, they contain chondroitin sulfate, so they have a little bit different functionality, but not much. Mast cells are similar to another type of cell that we find in the blood called basophils. Basophils also have lots of granules, and when we look at the blood, we're going to go into them in more detail. But basophils, by and large, stay in the blood, mostly. Mast cells, by and large, are in the connective tissue outside of the blood, so there's some differences there. And they come from different precursors. The reason that we kind of link them together a little bit is basophils also secrete heparin and histamine. Mast cells secrete heparin and histamine. So they have some similarities. They both work in inflammatory responses and as part of the allergic response. So even though they come from different sources, they're different sizes, their locations in the body are different, they do have some similarities that kind of link them together. When we look at the primary and secondary mediators, these are the chemicals that are in the granules. And we've mentioned histamine. Histamine is part of the inflammatory response. It makes our nose run and our eyes water and all that kind of thing. It's also part, of course, of the allergic response. So if we're allergic to pollen or what have you, and we get that, we might take an antihistamine to try to dampen that down a little bit. However, we have relatively benign allergies. They make us uncomfortable and our eyes itch and our nose run, but we can also have a severe allergic reaction, which is called anaphylactic shock. This is an extreme overreaction to the body's inflammatory pathway, where something triggers this overreaction and can result in such extreme vasodilation and increased permeability of the blood vessels that the blood pressure drops and can even be fatal. A blood pressure drop that is so low, it can be fatal. So some people are prone to these anaphylactic shock attacks. Well, here's heparin. This is an anticoagulant. Eosinophils are in the blood primarily, but also in the connective tissue. They travel in the blood, but go in the connective tissue. And of course, the blood is connective tissue, so I'm talking more about connective tissue proper here. Eosinophils work predominantly against parasites, worm infection, but they also have a little bit of role in the allergic response. Neutrophils are the most common white blood cell that's found in the blood, and it can also go into the connective tissue proper, and they are primarily involved in phagocytosis of bacteria and dead cells. They're usually the first white blood cell to reach the site of inflammation. So what are these two, ECF and NCF? These are chemotactic attractions for eosinophils and neutrophils. So these attractants are released from the mast cells, and they attract these white blood cells to the inflammatory site. Aryl sulfate is a negative feedback. It inactivates leukotriene, limiting our inflammatory response. So it shuts it down. It starts to shut it down. And so does chondroitin sulfate. It inactivates histamine. And then the proteases, they increase inflammatory response. So all of these are going to be released from the mast cells. Secondary mediators, you take a membrane instead of a granule. So they're called secondary mediators because they're not released from granules within the mast cell, but they cause a phospholipids in the membrane to be converted into prostaglandins and leukotrienes and these other thromboxane A2. And they have these different functions: contraction of bronchial, vasodilation, increased vasopermeability, and then here's vasoconstriction. That's more of a negative feedback one. And so these are what the secondary mediators do. So we have lots of these that are released by the mast cells. Here again is a light micrograph showing the granules. This reminds us that these granules can show metachromasia, meaning that the stain that stains them is a different color than the actual color that the granules become. So they don't match. The color that the granules don't match the stain that stains them. In most cases, the stain color and the structure that it stains are the same, they match. But if it's metachromatic, then they don't match. And so that's what can happen. And a number of the granules in mast cells can have this difference. So wonder why our detectors was weirdly. Wonder if it was a little electrical surge or something. Didn't seem to bother the computer, but the projector. Now, if we have an anaphylactic reaction, it's also called type 1 hypersensitivity, after a first initial exposure to an allergen, plasma cells, which produce antibody or immunoglobulin, let's just remind ourselves that antibody, so protein, is the same as immunoglobulins. Antibody equals immunoglobulins. You may have heard somewhere along the way that we have different classes of immunoglobulins. So IG for immunoglobulin, we have IgG, and IgA, and IgM, and IgE, and IgD. Okay, the ones that are responsible for the allergic response is the the IgE. IgG are the most common, but IgE are the ones responsible for the allergic response. And they can bind to receptors on the mast cells called Fc receptors. When these IgE, immunoglobulin E, or antibodies bind here, once you've had an initial exposure, now these cells are sensitized, and if you are have this predisposition for this severe allergic reaction, additional exposure can cause a severe allergic reaction, possibly life-threatening. This is why somebody gets stung with a bee and they have a reaction, but then they may be primed that if they're stung by a bee again, they'll have a more massive reaction. Most people they could be stung by a bee multiple times in their life, and it's not gonna result in that massive overreaction. But if you happen to be sensitive, and it's probably related to some genetic factors and some other things, but you could be, it could be life-threatening. So somebody like that might carry an epinephrine pen, because if they're getting this severe anaphylactic shock reaction, they can inject the epinephrine from the EpiPen, and that will increase the heart rate and cause vasoconstriction so that it's life-saving until they can get into some more medical care. So that would be what's happening. Now, what happens with this overreaction? And it could be pollens, it could be insect venoms, it could be drugs, like some people are allergic to penicillin, for instance, or in serum, a number of things. So what happens is when the antigen binds again, and it has its complex here with the IgE, so here's the antigen, here's the IgE, it binds to this Fc receptor, it activates a series of events which causes too much release of these mediators, the primary mediators, from these granules. And so a number of them get released, and that can cause this overreaction. Now, if you have a mild reaction, that's just a regular allergic response. But if you have a massive reaction, then that is what could be potentially life-threatening. Macrophages, we've said, come primarily from monocytes. Monocytes, like all the blood cells, come from the active or red bone marrow. That's where they're made. Monocytes go into the blood, and then they can leave the blood, and when they leave the blood, they can fuse together to form larger cells called macrophages. And so they're part of the mononuclear phagocytic system. Mononuclear is the name because actually all the white blood cells only have one nucleus, but a number of the white blood cells have multi-lobes, or more than one. Eosinophils are bilobe mostly, basophils are bilobe, neutrophils have 2 to 5 lobes, and so they appear like they have more than one nucleus. But monocytes and lymphocytes just they don't have the appearance of more than one nucleus. So they call them mononuclear, and so the monocytes have that, and so they're derived from monocytes, so we call it the mononuclear phagocytic system. And they are going to phagocytize, and we looked at a number of these. These are all macrophages. They just have different names to some degree, and they have different locations where they work. As we go through the quarter and we look at some of these, we'll remind ourselves of some of these names in different regions, but that tells us about where they're from. Another category of white blood cell are the lymphocytes. The lymphocytes are the second most common white blood cells in the blood. The neutrophils are the most common. The lymphocytes are relatively small. The cell is mostly filled up with the nucleus, and we have different subcategories. The T lymphocytes get that name because after they're made in the bone marrow, they go to the thymus, so they're thymus derived, and the ones that can auto-react with our body get taken out, and only the ones that don't auto-react with our body are released from the thymus. And they are going to be part of what we call the cell-mediated response, where they will attack our own host cells which have been altered, for instance, by a viral infection, or in certain cases, they've turned cancerous. So if our own host cells are altered, they have a virus inside them or something, the T cells will come and attack them and destroy them. The B lymphocytes, they also come from the bone marrow, and they are going to be able to be activated to plasma cells that are actively releasing antibodies. So B lymphocytes can be turned into plasma cells that they release antibodies, and that's part of the humoral or blood immune response. Natural killer cells tend to not have any surface determinants like these two do, but they're going to act against the tumor cells. Here's the plasma cells. Again, they're derived from B lymphocytes, and they are going to have generally an eccentric nucleus off to the side and be larger than the lymphocytes, and lots of rough ER and ribosomes. They make all these protein antibodies, and they have often a prominent Golgi complex, which is often seen under light microscopy as a negative Golgi image. Here's a negative Golgi image. All of these light regions here is where the Golgi apparatus would be. It's usually quite close to the nucleus, but it's in the cytoplasm. And so the negative Golgi image is indicative that this is Golgi apparatus is there. We've mentioned briefly that these three white blood cells, they have granules in their cytoplasm, neutrophils, most common white blood cell in the blood. It phagocytizes and kills bacteria predominantly, and it's the first one to the inflammatory site. Eosinophils primarily work against parasites, but they also are part of allergic response. Basophils are mostly part of the allergic response. Basophils are very rare. Less than 1% of the white blood cells in the blood are basophils. Eosinophils are pretty rare too, about probably 2 to 3% are there. Now, skipping ahead, since we've gone through this, let's just look at some of the clinical considerations for the connective tissue. Scurvy, we mentioned a bit, is a deficiency of vitamin C. During the production, synthesis of collagen, we need some hydroxylation to occur, and the enzymes that are going to allow that to happen, we need a coenzyme of vitamin C for that to work. So if we don't have enough vitamin C to act as a coenzyme, we get unstable collagen, and this causes a number of problems in our connective tissue. You may be aware that sailors back in the day, they would go on these long journeys in these wooden boats and the sails and all that, and they wouldn't have enough vitamin C. They didn't have fresh food and stuff. And so they would get scurvy a lot. And so they finally figured out, hey, we need to do something about that. So they would carry along with them like a lime tree, citrus, and they would eat those. And of course, back in that time, the British Navy ruled the world, and so they called British sailors limeys eventually because they ate limes, but it helped with scurvy. So there was a reason for that. Now, of course, we don't have that issue, but there could be times if people really had a poor diet, they might get scurvy just because they weren't getting any citrus or vitamin C in anything. Of course, in this country and many others, some of our food is fortified with vitamin C, meaning you add it. It's not in there. And so you often fortify even orange juice or something with additional vitamin C and other things. But getting a balanced diet helps generally with these kind of issues. When we have wound healing, we have to reconfigure both the connective tissue that got damaged and the epithelial tissue that overlies it. And so we form a protein called fibronectin tracts, and fibroblasts come and get activated, and they start healing up that damaged tissue. And the epithelial can also be regenerated. This is maybe a good time to remind us that epithelial tissue is avascular. It doesn't have blood vessels. It has to get its oxygen and nutrients from diffusion from the underlying connective tissue. Connective tissue proper is very vascular, and we'll see that bone is very vascular. What we'll see, though, today a little bit is cartilage is avascular in the interior, so it doesn't have a blood supply. This is one of the reasons that cartilage can kind of wear out and become a little more brittle as we get older, and it doesn't have that regenerative power, whereas connective tissue proper and bone can regenerate, and epithelial tissue can regenerate quite well. Cartilage doesn't regenerate, and so if you tear your cartilage, you can't really regenerate it, or it's very limited in its regenerative ability. Ehlers-Danlos syndrome is a genetic disease. There's 13 subtypes. Some are much more severe than others. But it's a DNA problem with either transcription or translation in the process of making type 3 collagen. And you remember that's reticular collagen or reticular fibers. And so when you don't have enough type 3 collagen, the skin is thin, translucent. That means it's light kind of goes through it. It's fragile, easily bruised, often stretchy, so you can get joints that are dislocated. You could dislocate your shoulder or your fingers or things like that much more easily. Of all these 13 subtypes, type 4 is the most serious because it affects the blood vessels and the small and large intestine. And if these rupture, of course, they can cause death. So this is one that most of the others, the people that have this live a normal lifespan, and they just have to be a little careful about things. But if they have type 4, then that could cause them to have a shorter life. You can't even, if they have type 4, you have to keep the blood pressure lower than normal so it doesn't break their aorta, for instance. Edema means you have an increased abnormal tissue fluid volume. This could be due to a number of reasons. Congestive heart failure, you're not moving the blood around enough, and so it's causing too much pressure in the capillaries and fluid leaks out. Starvation, if you eat your own proteins, you decrease the number of proteins like albumins in the blood, and so you have less of an osmotic gradient, so fluid goes out into the tissues. That's why if you see pictures of children starving and they have a big belly, you say, you got a big belly. It's not fat, it's fluid in the interstitial fluid, because in desperation, they're starving, their body is digesting their own proteins, and the proteins in their blood, and so they're getting edema. Hay fever and asthma, we talked a bit about already, so, and anaphylactic shock. Obesity, after a certain time of age, usually we think about 2 years of age, you don't increase the number of fat cells. But under the age of 2, you can increase the number of fat cells.
after the age of 2 approximately. If you get fatter, it's because the size of your current fat cells get bigger and you get that. And so there is some evidence that babies that are overfed or fed an unhealthy diet might be more prone to obesity later in life because they have increased the number of fat cells too much. And so this would be hypercellular obesity. So, people that did give their babies, soda to drink or something, probably, or, they're not doing them any favors later in life, of course, or even at that stage. On the other hand, adult hypertrophic obesity is increasing the cell size and not the cell number of fat cells. All right, so that's we've looked at these images. I wanted to go through just a few more of those images. This is from the lab review slides that Trinity you were looking at for epithelial. This is on your canvas, but you're going to be hearing on the Friday audio lecture about cartilage and a pretty good introduction to bone, and then we'll finish up the bone next Tuesday. But I wanted to show us just a little bit of cartilage in lab today after we finish up the regular connective tissue, so that we get a little bit of a head start. First of all, this is our connective tissue. This is dense irregular. You've seen this slide before, but just to show it to you fairly quickly here. Remember that this is, I'll go ahead and make it a laser as Spencer taught me. This is artifact, right? Remember that that is a something that's due in the producing of the slide that isn't indicative of the actual structure found in vivo in living organism. And so it pulled away here. These are collagen fibers that are irregularly oriented and fibroblasts. So we'll be looking at that in lab later today. Here it is with, again, this irregular shape. This is in the dermis. We'll look at it in the dermis, connective tissue of the skin. This is regular and a tendon. Again, this is artifactually separated a bit. And here it is again with the ligament. Now, let's look at cartilage a bit. Now, again, we're going to be giving you quite an overview of cartilage in the audio lecture, but I wanted to show you the slides of the 3 main types of cartilage, so we're starting to get a little view of it, and I'd like to show it to you on the monitor as well today in lab. The first thing to notice is, as we mentioned a little earlier, cartilage, the main part of cartilage is avascular. It doesn't have any blood vessel supply. It has to get all of its nutrients by diffusion from the underlying connective tissue proper, for instance. Cartilage is in between connective tissue proper and bone in that it gives structural support, but it has more flexibility than bone. Bone is more stiff and strong. Cartilage has a little bit more flexibility. And we find cartilage throughout the body. We find cartilage at the ends of the long bones. We find cartilage a little in our nose and in our ears and in our lining around our trachea, our airway, and in our epiglottis, that little flap that comes over the larynx, trachea. We find it in the larynx, the voice box. We find it where the pubic bones come together. We find it between the vertebrae, the intervertebral disc. We find it in lots of different places. Functionally and somewhat Structurally, we can divide the types of cartilage into three major subtypes. And again, this is gonna be in the audio lecture and in the notes, but they're hyaline cartilage, and they're elastic cartilage, and they're fibrocartilage. Hyaline cartilage being the most common, fibrocartilage being the rarest, but they're all three present. Hyaline cartilage, one of the things it does, it makes up the little models of the long bones that we turn into the long bones. When we're in the embryonic and fetal state, you start making bones as little hyaline cartilage models, and then you turn it into bone. Okay? Hyaline cartilage is the articular cartilage over the ends of the bones so that they don't rub together. They're nice and smooth and like that. And then we have hyaline cartilage around the trachea, for instance, a number of different places. Elastic cartilage is going to be more flexible, elastic like in the ears and in the nose, places like that. It's in the epiglottis that kind of flaps up and down. Fibrocartilage is part of the cartilage between our vertebrae and between the pubic symphysis, where the pubic bones come together. Those are all fibrocartilage. All cartilage and all bone have little spaces within the cartilage called lacunae. And again, that'll be in your notes, your regular notes, but I just want to let you hear that name. Lacunae are the little spaces in there, and we have cells in those little spaces. One of the ways we identify cartilage is by the presence of lacunae, or these spaces. If it has lacunae, it's either going to be cartilage or bone, so that helps us. This is a trachea. You probably recognize, even though this is low magnification, that this is our pseudostratified ciliated columnar epithelium that we're tracing along here. This is connective tissue proper. This would be loose connective tissue here. You see a number of blood vessels in cross-section. Well, this is cartilage right here. Here's our cartilage coming around like here. The edges of hyaline cartilage and elastic cartilage have this edge here and right here. That is called the perichondrium. Cartilage, we use the word chondro. You may even remember this when you're classifying sharks and rays, because they have cartilage instead of bones. So, chondro. So, we're going to have cells called chondroblasts and chondrocytes, for instance. Well, this tissue around the edge of the cartilage is called or around chondrium. So perichondrium, perichondrium. The perichondrium is vascular. It can have blood vessels. But the interior of the cartilage is avascular. It doesn't have any blood vessels. When we look at a higher magnification, this is still the trachea. Here's our pseudostratified ciliated epithelium. Here's some loose connective tissue with blood vessels. You can see the red blood cells in there. This is our cartilage. This would be perichondrium from about here to here. This would be perichondrium on the other side from about here to here, merging into dense regular connective tissue. And this would be the rest of our cartilage. All of these spaces in here, like here and here and here and here and here, those would be the lacunae. On the edge of the cartilage, we have chondroblasts. That's the less mature form, and they can lay down new cartilage. Now, once the cartilage is made, you don't really regenerate it anymore, but they lay it down and move it. When you're a little bit deeper and into the lacunae a little bit more, these cells are now called chondrocytes. They help maintain and give nutrients and things to the cartilage. You can also have groups of these lacunae and chondrocytes. These are called isogenous groupings, because they're all derived from one cell, stem cell. Hyaline cartilage in the matrix outside of the cells, we really don't see the fibers too well. There's collagen fiber there, type 1, but we don't really see it too well. This is hyaline cartilage. Here's our perichondrium. Here's our chondroblasts that are on the edge here, and then the chondrocytes. And then we have these isogenous groupings clumping together. The ground substance, the extracellular material here, looks rather smooth. You can't really differentiate the fibers particularly well. Here you can see fibers in the perichondrium, but here you're not seeing fibers too well. And so that's a characteristic of hyaline cartilage. This shows you a chondrocyte in a lacunae. I'm going to go fairly quickly here. This is an elastic cartilage, and it does have a perichondrium, and then these lacunae, and again, chondroblast on the edge, chondrocytes. What's different? When we stain it, counter stain it with the Van Giesen stain or something like that, we'll see these elastic fibers that are black here, and they are going to be giving it a little bit more flexibility. This happens to be a slide of the epiglottis, but from this part right here, from here to here and here to here, that's the elastic cartilage within it. And it can bend a little bit more than hyaline cartilage. Interesting quick note, because we've counterstained this with Van Giesen, notice the red blood cells in the blood vessels are green. So don't get fooled by what's that green stuff in there. That's the red blood cells. It's just a different stain. Makes it look kind of weird. This is a higher magnification. And see the spaces? That's the lacunae. And out here we see the chondroblast. Here's the perichondrium. But chondrocytes here. Look at the elastic fibers. And that helps us know that we've got elastic cartilage here. Here it is again. Fibrocartilage, this is a bone, a vertebral bone, and here's a bone, and hyaline cartilage on its surface, but then between you have the intervertebral disc, and that's fibrocartilage. Fewer cells, and you can generally see the fibers more clearly. So we're seeing fibers there. So that's fibrocartilage. Okay, so I wanted to give you that a bit of orientation. I know that that was quick and incomplete, but in lab today later on, we'll look at those three types again. We'll skip embryonic for now, but we'll look at those three types, and that'll give us a little bit. And then when you decide I'm ready to listen to that audio lecture that's sort of due on Friday, it will give you a more lengthened orientation about cartilage, and then it'll give you about half the material on bone, and we'll finish up the rest of the material on bone Tuesday when we come back at 2 o'clock on Tuesday, and we'll look at, again, the cartilage and the bone, all those slides, so we won't have to meet next Wednesday, the day of the test. We'll just meet for the test during lab time. So that's why we're pushing a little bit here. Okay. So we'll see you at 2 o'clock. Remember, quiz first in room 201, Dr. Clark. Then we'll go down, we'll finish up the connective tissue and the connective tissue proper. And then we'll look at the three, quick look at the three kinds of cartilage.